Okay, so now we've examined stem cell research and the ability to use somatic cell nuclear transfer for the possibility of making embryos for stem cells. And I position that as a backdrop to cloning. So we recognize from the first lectures that most of the DNA in a cell is in its nucleus, shown in this diagram in blue. So we also, and remember, not all. There are genes in DNA in mitochondria. And in plants, there's also DNA in chloroplasts. So there are genes associated with the mitochondria, and we'll just keep that in mind for a minute now. But most of the DNA in a cell is present in its nucleus. So all of the information for each protein in a cell, whether it's in its nucleus or its mitochondria, is in its DNA. So therefore, the majority of proteins that are produced in each cell are in its DNA. And remember that every time a cell divides, that DNA is copied nearly exactly. So every single one of your cells has a nearly exact copy of your DNA. This becomes relevant for all kinds of things that we'll talk about later, from forensics to now cloning. But cloning is an intense example biologically of that fact. So all the information that codes for a complete individual is present in every cell of your body. So by taking any individual cell and removing its nucleus, we now have the information that is specific for that individual and that organism. Also, we would have the ability to add foreign DNA by transgenes. So the ability to both clone and genetically engineer animals is currently very functional and reproducible. So we can both genetically engineer animals as well as genetically clone them, and we can also use fertilization technologies similar to human fertilization technologies for animal breeding purposes. So breeding of animals now can be accelerated using genomics and genomic technologies in a way that we couldn't do before, accelerating conventional breeding of animals. And now we have, similarly to plants, the ability to genetically modify animals and clone them. So how to clone? How do you actually clone something? Well, a donor cell, a somatic cell, say a cell from your earlobe, could be used. Anything with a nucleus in it. That would not be a red blood cell. So shown here in the upper right, those are nuclei, which could be taken up into a small micropipette needle. And on the left, you have a oocyte, which has had its nucleus removed. An enucleated oocyte then would be the donor. The somatic cell nucleus, which is a diploid nucleus, contains all the information for that organism, is then injected into that unfertilized egg. The structure that's shown off to the far left is a micropipette, which applies a slight suction on the egg to hold it into place during the injection of the needle. So by this way, Dolly was created as the first cloned sheep. So again, it's injecting a 2N nucleus into an enucleated egg and then stimulating that to divide. So here's some actual micrographs showing nuclear transfer and what that might look like underneath a microscope. In some cases, the injected oocyte is stimulated to divide by an electrical pulse and then implanted. So an organism can be cloned then by transferring a somatic cell nucleus, hence most of its DNA, into an unfertilized enucleated egg cell. This demonstration is dramatic and won worldwide acclaim, where it strikingly shows that all the information that codes for an organism is in its DNA, and that all of the DNA is the same in every cell. Hence, I can take a liver cell as a donor, an ear cell, a mammary cell, etc., and reiterate that same genetic program. I think it is important to recognize that that enucleated egg cell has its own mitochondria, and hence then it's that mitochondria that is providing those mitochondrial gene functions against the implanted DNA. It's a small little caveat, perhaps, but I think it bears recognition. So we could genetically engineer cloned animals. Why would we want to do this? Well, you could do this for all kinds of reasons. You could do it, say, to increase muscle mass, make muscle mass grow faster, if that's your goal. Yield, for example, in poultry, swine, and cattle. You could have mammals make vaccines as a perpetual source of a vaccine. If it were difficult to make, you could have one transgenic animal continually making that vaccine or that antibody, or for that matter, other high-value proteins. There have been examples where goats, for example, have been genetically modified to make spider silk. You might think, well, that's pretty weird. But actually, spider silk has some qualities structurally that could be very useful. Some people have said it's stronger than Kevlar, bulletproof vests and so on, lightweight materials that might be functional for all kinds of purposes. And so they might be expensive to make at the outset, but one goat could make a lot of milk and hence derive spider silk or other high-valued proteins. You name the protein, and it probably would be determined whether or not it was economically significant in order to do it that way. But if you think of something that's made in small quantities that has high value, your best approach might be to put it into an animal and derive the protein that way. So genetically modified somatic nuclei can be implanted into unfertilized egg to create a genetically modified clone. And this has been done in all kinds of animals, as I'll show you in a minute. To the lower right here is an example of a cloned pig, produced by the same people, by the way, that brought us Dolly. And in fact, clones of clones of clones have been produced. So you can reiteratively clone animals. And in this exercise, they were specifically interested in genetically modifying out the proteins that are responsible for immunorejection, so that they could make a line of pigs that would serve as human organ donors, so that you could have a kidney pig that was humanized and that it would remove the glycoproteins that are recognized by the human immune system, allowing then for these organs to be useful in xenotransplantation. So you can use transgenics and cloning in tandem. So these could be useful for making drugs in animals that are also of high value and have a long-term source of this. What exactly is cloning? We've already used that word when we've described cloning a gene, where cloning a gene should not be confused with cloning an animal or an organism. Cloning a gene refers to the process of copying a fragment of nucleic acids, primarily DNA, and copying that many times in a host bacterial cell or something like that. Whereas cloning in its reproductive sense is a process whereby all the members are directly descended asexually from a single organism by various ways. This would count then for vegetative propagation of plants. Your grandmother's African violets would count as clones in that regard, but also the animals that have been cloned by somatic nuclear transfer technologies. But again, I want to point out that in any of these cases, it demonstrates that all the information that's required for a you is in every cell in your body. Is it you exactly? No. It's a genetic twin. Are genetic twins absolutely identical? Not quite. But it does demonstrate that we can physically move that information via the transfer of simply the nucleus. We also know now the genomes of many organisms. And this ability and all of this information becomes confluent in the ability to genetically modify as well as clone these organisms. So in principle then, the ability of cloning not only relies on the fact that all the information in every one of your cells codes for a you, I mean a person, you, all cells are derived from a zygote. And all cells then have the same genetic makeup by the virtue of the fact that DNA replication occurs prior to cell division. And all have the potential to develop into an individual. So that by restarting the program into an unfertilized egg from a skin cell, no longer will a skin cell, just a skin cell, beget. Follow? Instead, you can trigger this thing to restart the genetic program to make an entire individual. It'll look like an identical twin of you, however, it will be restarted. So these science fiction notions that there would be, you know, lots of you running around fully matured that you'll talk to at this, you know, it's kind of wacky notion, right? But I think you can see how this goes. So in animals, cats have been cloned. This is an example of the first cat that was cloned. They named it Cece, I think for copycat. And it was conducted by a group in Texas that wanted to set up the business of cloning pets. Their company was called the Savings and Clone, and they have gone out of business because it was economically prohibitive, but Cece was produced by that group. Mules, a race mule was produced at the University of Idaho as a clone. You might say, why would you do that? Well, if you had a prize mule, there's no other way that they can reproduce. So they cloned the mule. Horses have been cloned, and I think there are rules by the Thoroughbred Racing Association to prohibit this kind of technology. Would you want, you know, a whole bunch of, what was the one that just almost won the Triple Crown? I can't remember. But, you know, you wouldn't want a whole bunch of that one crossing the finish line. And you might want to say, would they really all cross the finish line nose to nose? No, just the way genetic twins wouldn't do that, right? The variation exists because of that variation in genetics plus environment factor and other small factors such as single nucleotide polymorphisms. As I mentioned, pigs have been cloned for all kinds of purposes, including xenotransplantation, but humanized pigs for experimentation have been developed and are very useful even for examining things like the effects of the microbiome. You can make a humanized pig and transfer into that pig human microbes and watch how these are colonized and affect that organism. Here is a photo of a friend of mine, Dr. Jerry Yang, who passed away a few years ago. Actually, he lectured to this class, and he He said that it was a privilege to be here, and this was his first public speaking engagement after his last brain surgery. He died of actually progression of salivary gland cancer, which is sort of ironic because he was in the Department of Regenerative Biology at the University of Connecticut, and he was the first person to clone cattle. And you can go over to Horse Barn Hill behind the building for agricultural biotechnology on Storrs Road and see the outcome of Jerry's research. So the cloned cattle are still over there on Horse Barn Hill. A remarkable contribution by a remarkable man. And there are some of the Yukon clones. I can't remember the donor's name, but I like to show this picture because here are the 4 clones shown here, and you can look at if you look carefully at the pattern of the color markings on their faces, you can see, in fact, slight variations. So they are not, in fact, all identical. So sheep obviously have been cloned. Everybody knows that since Dolly. Those experiments have been repeated. And I don't know if you heard this controversy that Dolly died prematurely, and everyone was wondering if the cell from a mature tissue already has its clock ticking, so to speak, and that Dolly died prematurely. This has not been substantiated in subsequent experiments. There is maybe something to this in terms of how telomeres age in chromosomes and setting up a clock. And the studies of telomerase and telomeres have focused more on the aging process, but it turns out that this was, owing to a large sample size of Dolly. As you look at this, this doesn't seem to be borne out, say, in the clones of clones of clones of clones of clones of pigs, for example, or mice. And now we have recent examples that rhesus monkeys have been cloned, goats. So the list is growing. So many animals can now be cloned. I would propose that this technology can be widely applied. Have all animals been cloned? No. That might be merely a matter of elbow grease. What would be physiologically the barrier between applying this, I think, is really a matter of finding the correct parameters experimentally. So, for example, could this be done to humans? I cannot understand why there would be a biological barrier to this. And what would make humans so special that this application could not be more widely applied? Primates, the cover of Cell here demonstrating chimeric monkeys, I think demonstrates its application in primates. Pretty cute, huh? Really? Okay, science fiction. Will we ever see dinosaurs? These days, would you ever say never anymore? You're hard pressed to say, well, if you had the appropriate DNA and synthetic biology approaches, given this kind of technology, how long will it be before we can make extinct animals? In fact, extinct animals have been made already. A bantang was cloned from a cryopreserved tissue and implanted into a bovine surrogate, and there are now extant bantangs, where previously they were extinct. How widely can this be applied? I think this is a great application, not only for maybe recovery of extinct organisms, but certainly preservation of endangered ones. This shows that the cloning of plants goes back even further to 1958, when Stewart did this at Cornell, demonstrating that totipotency is possible in plants, but that this now has been shown through the work of Ian Wilmot and people like Jerry Yang, that this is possible in animals as well. Okay, is it possible to clone animals? Sure, it's been done. This is not science fiction, it's reproducible, it's evidence-based science. We can genetically engineer them. Can we clone humans? Well, you can see how long will it be. Is there a reason to do this? You can say, well, geez, we've got plenty of humans so far. Fertilization clinics, though, do quite well. But on the other hand, would there be a reason that we would genetically engineer humans? What if I told you that the best way to cure cancer might be to eliminate it genetically from the human lineage? Start with an unfertilized egg and simply delete oncogenes out of that pre-fertilization. Is that apparent? What about Parkinson's disease? By genetic testing, we could analyze that your offspring-to-be will have Parkinson's disease. Could we just take that out genetically by genetic modification or enhancement of certain genes? You look skeptical. But certainly, if this can be done, you're on now this ethical slope of, is this the best way to cure cancer in humans rather than deal with it when it happens, just preemptively remove it as a possibility? What about severe combined immunodeficiency? What about beyond therapy? Okay, you could justify this in terms of saying, let's take out cancer, let's take out diabetes. But what about height? You know, Mrs. So-and-so, to be a professional basketball player, you know, you might want to have your son to be over 7 foot or so. These days, 8 isn't even sufficient, you know. So I don't know, you know. Beyond human therapy is a question. How would this happen? Well, you can imagine even with the archaic techniques that are around today. Humans could be cloned, say, from taking amniocentesis cells, which are fetal cells sloughed off, and that could be sampled from the amniotic fluid. These contain nuclei. One of those nuclei could be withdrawn, genetically tested, genotyped, right down to the single base pair, and diagnosed. If we found out that we wanted to replace something, a gene could be implanted into that nucleus and reimplanted into an unfertilized egg, and the pregnancy then could be restarted with 1 gene or 2 genes replaced or manipulated. Think back about the Tay-Sachs dilemma. If you knew the baby-to-be would suffer a miserable short life, would some people opt to do a corrective genetic surgery? Do you think we should genetically engineer humans? Will we? Maybe the question really should be changed to, what will you think when we? Would you like to clone yourself, if that were possible? Is that the son or the daughter you'd like to have? People in my class responded positively to this notion. So the possibilities of human cloning are really at our technological doorstep. I don't think that ethically we are prepared for this, not by any means. We can't even decide over pro-life or pro-choice. But technologically, this is certainly within the time span of, say, less than a half of a Chinese dynasty. To clone or not to clone? That's not really the question. It's more like, what will you think when we? Okay, we'll take a break.